3D microvascular networks represent a cutting-edge fusion of tissue engineering, microfluidics, and biomimicry. These innovative systems create intricate networks of tiny channels that closely resemble natural blood vessels, enabling the delivery of nutrients, oxygen, and biochemical signals throughout engineered tissues. By designing microfluidic channels with dimensions comparable to capillaries, researchers can mimic the complex vascular architecture found in living organs, a crucial step toward building fully functional tissue constructs. One of the most impressive approaches involves using advanced 3D printing and soft lithography techniques to sculpt these microvascular networks. For example, engineers can print sacrificial materials in a complex branched pattern, embed them in a hydrogel that serves as the tissue scaffold, and then remove the sacrificial template to leave behind hollow, interconnected channels. These channels are then lined with endothelial cells, the same cell type that forms the lining of natural blood vessels, to create a living, dynamic vascular network. This process raises an intriguing question. Can we one day fabricate entire organs with fully functional vascular systems that integrate seamlessly with a patient's own blood supply? The benefits of these 3D microvascular networks extend far beyond structural mimicry. In tissue engineering, one of the biggest challenges is ensuring that thick, engineered tissues receive adequate blood flow to remain viable. Without a properly functioning vascular system, tissues can suffer from hypoxia and nutrient deprivation, leading to cell death. By incorporating microfluidic channels that act as blood vessels, scientists can maintain tissue viability over larger volumes, opening the door to the creation of more complex and functional tissue constructs for regenerative medicine and organ transplantation. Moreover, these networks also provide an excellent platform for disease modeling and drug testing. Researchers can use 3D microvascular networks to simulate conditions such as tumor angiogenesis, where new blood vessels form to supply a growing tumor, or to study the dynamics of blood flow in response to various therapeutic agents. The ability to control and monitor fluid flow within these channels offers a powerful tool for understanding how vascular systems respond to disease or treatment, ultimately leading to more targeted and effective therapies. As we look to the future, several compelling questions arise. How can we further refine the design of these microvascular networks to more accurately replicate the dynamic, adaptive nature of natural blood vessels? Could we eventually integrate sensors and real-time monitoring systems within these networks to track tissue health and function continuously? The ongoing advancements in 3D microvascular network technology not only deepen our understanding of vascular biology, but also push the boundaries of what is possible in tissue engineering and regenerative medicine. 3D microvascular networks are engineered systems designed to replicate the intricate network of blood vessels found in living tissues. They employ advanced microfluidic techniques to create channels that mimic natural capillaries, arterioles, and venules. These networks are essential in tissue engineering as they enable the transport of oxygen, nutrients, and waste products. The design aims to closely simulate the physical and biological environment of human vasculature. This innovation helps bridge the gap between traditional cell cultures and actual human physiology. Scientists design a scaffold embedded with tiny, interconnected channels that mimic the branching pattern of blood vessels, allowing lab-grown tissues to receive the necessary nutrients for survival. Imagine constructing a miniature city with an elaborate network of roads and highways that deliver essential supplies to every neighborhood, ensuring the city runs smoothly. The design of 3D microvascular networks is guided by the geometric and functional properties of natural blood vessels. Engineers utilize computer-aided design (CAD) software to simulate the branching hierarchies observed in human vasculature. The goal is to ensure that the engineered channels can efficiently transport fluids in a manner similar to natural blood flow. This process involves careful planning of channel dimensions, angles, and interconnections. Such design principles are crucial to achieving a realistic and functional mimic of the human vascular system. Researchers create a CAD model that features gradually narrowing channels, which emulate the transition from larger arteries to tiny capillaries found in the human body. Think of planning a city's road system where major highways branch into smaller streets, ensuring every area is well connected and easily accessible. Various fabrication techniques such as soft lithography, 3D printing, and photolithography are employed to create these microchannels. These methods allow for precise control over the dimensions and geometries of the channels. Biocompatible materials are selected to support cell growth and mimic the mechanical properties of natural blood vessels. The fabrication process is carefully optimized to ensure the channels are robust and functional. This technological advancement is central to replicating complex vascular networks in a controlled laboratory setting. A research team uses 3D printing with biocompatible hydrogels to layer complex, branching channel networks that replicate the structure of human blood vessels. Imagine using a high-precision printer to create a detailed model of a city's road system, where every street and alley is accurately reproduced to ensure the model functions just like the real thing. Biomimicry in microvascular networks involves designing channels that closely resemble the structure and function of natural blood vessels. The process replicates key features such as elasticity, permeability, and branching patterns. This approach helps in creating an environment that supports natural cellular behavior and tissue integration. Mimicking these properties allows engineered tissues to respond to stimuli in a manner similar to their in vivo counterparts. The ultimate goal is to ensure that the engineered vasculature is not only structurally accurate, but also functionally active. Researchers develop microchannels with flexible, responsive walls that simulate the pulsatile nature of blood vessels, adjusting to changes in pressure and flow just like natural vessels. It is like designing a synthetic fabric that not only looks like natural leather, but also stretches and breathes in the same way, making it almost indistinguishable from the real material. Integrating 3D microvascular networks into tissue engineering scaffolds is key to supporting cell survival and function. These networks facilitate the delivery of essential nutrients and oxygen while removing metabolic waste from cells. The integration creates a dynamic environment that encourages tissue maturation and regeneration. It allows for the development of thicker and more complex tissues that better mimic native organs. Such integration is crucial for the advancement of regenerative medicine and the creation of transplantable tissues. In liver tissue engineering, microvascular channels are embedded within a scaffold to sustain hepatocytes, resulting in a model that more accurately reflects liver function and metabolism. Imagine installing a sophisticated irrigation system within a garden that ensures every plant receives the right amount of water and nutrients, promoting robust growth and health. Dynamic fluid flow is a critical aspect of mimicking natural blood circulation within microvascular networks. The engineered channels are designed to replicate the pulsatile flow and shear stresses found in real blood vessels. Controlled fluid dynamics ensure that the cells experience physiological levels of mechanical stimulation. This dynamic environment is vital for maintaining cell viability and promoting tissue development. It also allows researchers to study the effects of fluid flow on cellular behavior under near-physiological conditions. A microfluidic device is programmed to pump culture medium in pulses through the network, replicating the rhythmic contractions of the heart and providing mechanical cues to the cells. Imagine a water fountain that not only delivers a continuous stream of water, but also mimics the natural rhythm of a heartbeat, creating a lifelike and soothing display. 3D microvascular networks offer a realistic in vitro platform for modeling diseases and testing new drugs. These systems replicate the complex interplay between cells and blood vessels found in human tissues. By simulating real physiological conditions, they improve the predictability of drug responses and disease progression. This capability reduces reliance on animal models and enhances the relevance of preclinical studies. Ultimately, these models help accelerate the development of safer and more effective therapies. A tumor-on-a-chip model incorporates microvascular channels to study the interactions between cancer cells and the surrounding vasculature, allowing researchers to assess how drugs affect both tumor growth and blood vessel function. It's like creating a detailed simulation of a city's traffic system to test how new road designs or traffic signals will perform before they are implemented in real life, ensuring optimal functionality. Replicating the complexity of natural vascular networks in a controlled lab setting presents significant challenges. Issues such as material compatibility, long-term stability, and precise control over fluid dynamics must be addressed. Researchers continuously innovate to overcome these obstacles by developing new materials and refining fabrication techniques. Balancing structural accuracy with functional performance remains a primary focus. These challenges drive ongoing research and innovation in the field of tissue engineering. Scientists are experimenting with novel hydrogels that improve cell adhesion and maintain the flexibility required for dynamic fluid flow, addressing the challenges of replicating natural vessel behavior. Consider the challenge of constructing a model airplane that not only looks realistic but also flies reliably. Engineers must carefully select materials and design methods to balance aesthetics with performance. The future of 3D microvascular networks lies in their further integration with stem cell technologies and personalized medicine. Advancements are expected to lead to fully vascularized, transplantable tissues and organs that closely mimic human physiology. This progress will significantly improve the outcomes of regenerative therapies and reduce issues related to tissue rejection. Ongoing research is focused on enhancing the scalability and functionality of these networks for clinical applications. The impact on regenerative medicine could revolutionize how diseases are treated and organs are replaced. Researchers are combining patient-designed V cells with custom-designed microvascular scaffolds to create transplantable tissues, paving the way for personalized organ replacement therapies with reduced risk of immune rejection. Imagine upgrading from a generic mass-produced car to a fully customized vehicle built exactly to your specifications, ensuring optimal performance and compatibility with your unique needs.